Good afternoon. My name is Rona Hartzano, and I am a professor of otolaryngology, head and neck surgery, and an otologist at the University of Maryland School of Medicine, Baltimore, and chair of the ANS Research Committee. I'd like to welcome you to the ANS Research Grant session, and I would like to start by thanking our committee members, Michael Kluth, Kristen Dean, Benjamin Crane, Susan Amet, Aaron Remenschneider, and Matthew Croson, who all participate in ranking the research grants. As a reminder, the ANS Research Grant is a $25,000 grant for 1 year to encourage and support academic research in sciences related to the investigation of otology and neurotology, and prepare clinician scientists to early career research awards. The applications are due March 1, 2022. Our funding line is quite good, and we provide very instructive feedback that helps applicants prepare for K Awards. Before we go to our research presentations, this year we put together an AOS-ANS protected time and compensation study to look for barriers in developing research careers in otology and neurotology. The survey takes merely 5 to 7 minutes to answer. You will soon have the link in the chat. If you have not answered this survey, please do take the time to answer it. It is still open today and tomorrow. And now we will transition to our ANS Research Grant presentations. We will have 2 presentations from 2 awardees. The first one is Dr. Courtney Volker, that will talk about in vivo neuronal mapping of the auditory pathways in pediatric patients with congenital unilateral sensory neural hearing loss and those with normal hearing. And she will be followed by Dr. Tatiana Correa about effects of delivery approach on helper-dependent adenovirus transduction of guinea pig cochlea. At the end of the presentations, there will be times for Q&A. So I thank you for your listening, and I look forward to the presentations.
Great, thank you, Dr. Hartzano, and I wanted to thank the ANS particularly for this grant, and especially the committee members that took their precious time to review all of the grants. I have nothing to disclose. So imaging technology has really made exciting advances recently. This study used MRI imaging techniques called Diffusion Mri to map neuronal connectivity in the auditory pathway of children during development. Diffusion-based tractography techniques can reconstruct white matter tracks to depict trajectories of connectivity in the living human brain and correlate structural inter-individual differences with behavioral performances. Diffusion MRI is based on the capacity of MR imaging to measure the extent and direction of water molecule diffusion within the central nervous system tissue. Water molecule diffusion in white matter is hindered by axonal cell walls and myelin sheaths and is therefore ionotropic, occurring preferentially along directions parallel to fiber track orientation. Diffusion MRI processing protocols are used to identify axonal bundles, estimate axonal orientations, obtain objective measures of their directionality, myelin status, and other tissue properties. Diffusion MRI is an increasingly important clinical and research tool and has been used widely in conditions such as stroke, traumatic brain injury, schizophrenia, dementia, and visual impairment. So much of what we know about the development of the auditory system comes from animal studies and functional MRI imaging. Studies of human auditory pathways during development have mostly focused on histological techniques. The goal of this study was to characterize the magnitude and timing of auditory pathway microstructural maturation in normal hearing children, and to this end, a hybrid streamlined tractography approach to reconstruct the various components of the ascending auditory pathway during development using the multi-tensor model was developed. The ascending auditory pathway was separated into a lower pathway and an upper pathway. The lower pathway was from the cochlear nerve to the inferior colliculus, and the upper pathway from the inferior colliculus to the auditory cortex. We started with a CMIND data set, which stands for Cincinnati MR Imaging of Neurodevelopment. This is a pediatric neuroimaging research consortium between Cincinnati Children's and the Laboratory of Neuroimaging Lonnie at USC. And its purpose is to characterize normative, structural, and functional brain development across a wide age range from birth to adolescence. There were 104 typically developing children from 0 to 18 years of age. The metrics that were measured were the neurite density index, which is a neurite orientation, dispersion, and density imaging, or NODI parameter, as well as FA, medial diffusivity, radial diffusivity, axial diffusivity, all which are DTI measures. Analysis was done in two ways. First, by looking at age-related changes across the microstructure of the whole tract analysis. Metrics were averaged across the core of each tract to reduce partial voluming with other fiber bundles. Age-related metrics were then assessed with nonlinear growth models, and the age at termination maturation was defined at the age at which the metric reaches 90% of the asymptote. Regional developmental patterns of diffusion were then assessed across a long track analysis. Each track was partitioned into equidistant segments, and the developmentally timing of each of these diffusion metrics was calculated at each cross-sectional vertex. Whole tract analysis showed that the exponential growth curve provided the best fit model, and overall, the lower auditory pathways reached terminal maturation earlier than the upper auditory pathways. The long track maturation also confirmed these findings, that the lower track matured earlier than the upper track. The next part of the analysis then looked at the laterality index, which was calculated and showed that the left auditory pathway developed earlier than the right auditory pathway. So in conclusion, the auditory pathway microstructure followed differing maturation time courses. The auditory pathway maturation is not uniform. The lower auditory pathway reaches adult-like levels earlier than the upper auditory pathway in a bottom-to-up pattern. And interestingly, also there is a left lateralization in the development, which matches the language development patterns. So findings from this study confirm histology and functional studies on development of the auditory pathway. This is very encouraging to use this as a confirmatory technique. This is a reliable technique for studying the microstructure of central pathways. It can be applicable to our field of hearing and balance, and to evaluate various patterns of hearing loss in our different patient populations. COVID did delay me scanning different groups of patients. We are starting to scan more pediatric patients with congenital unilateral profound sensory neural hearing loss, and I'm grateful to the ANS for giving an extension to our grants to do such that. I want to thank my collaborators, and I want to specifically highlight Laurel Fisher and honor her. Many of you know her in this group, and she died of glioblastoma during this project. I also want to acknowledge Art Toga, Stephanie Bodison, Kristen Lynch, and Ryan Cabine at Lonnie. Thank you very much.
Thank you very much, Dr. Volker. For the audience, please put your questions in the chat. And our next speaker is going to be Dr. Tatiana Correa. We'll talk about effects of delivery approach on helper-dependent adenovirus transduction of the guinea pig cochlea.
Thank you. And I'd like to start by thanking the ANS Research Committee and the ANS administrators who have put in all the work to make this work possible. So today I'll be presenting some of the work I've done in the last 2 years of dedicated research that I did with this funding, looking at the effects of delivery approach on helper-dependent adenovirus transduction of guinea pig cochlea. I have no disclosure, so we can dive right in. So as we all know, there are no treatments to preserve and or repair sensory neural hearing. However, increased understanding of the molecular pathways at work in sensory neural hearing loss has led to the next frontier, which is developing molecular therapies to preserve or restore hearing. So people are looking at a number of different things at this time, including stem cell therapies, growth factors, gene therapies, just to name a few. And at the forefront of potential options has been gene therapy using viral vectors to deliver genetic sequences to the inner ear. And of the viral vectors that people have looked at, adeno-associated virus has been the most studied thus far. And it's a great vector in terms of toxicity, and it's probably the most studied vector for the whole field of gene therapy, both in all parts of the body. However, one of the biggest drawbacks for this vector when it comes to the inner ear is the size. So the carrying capacity is a little bit limiting for the complexity of the different targets that we would want to address if trying to address sensory neural hearing loss. Because of this, we looked at a potential different vector that we could use for hearing loss, which is helper-dependent adenovirus. So helper-dependent adenovirus is basically an adenovirus particle that is devoid of viral coding sequences, which makes it non-toxic. And then furthermore, it also allows us to carry larger quantities of genetic cargo, which is exactly what we would want to overcome this hurdle of needing to carry large amounts of genetic material to address sensory neural hearing loss. And so the goal of our project was to characterize helper-dependent adenovirus distribution and transduction in the guinea pig cohort. And the reason why, can go back one, yeah, perfect. So the reason why we needed to do this was because the helper-dependent adenovirus vector has been less studied. We don't really know where it's going to travel when we deliver it to the inner ear. And so that's why we decided to pursue this specific project. And so the way we did this was we injected helper-dependent adenovirus with a type 5, which is the most common adenovirus vector. And we picked this because it has a well-established cell entry receptor. And then we also put a ubiquitously expressed CMV promoter to drive a fluorescent reporter protein. And then we injected the guinea pig with the vector via different approaches. And then after 7 days, we sacrificed these animals and then did immunofluorescence staining and imaging. So the 3 different ways we approached this was by looking at delivery through the scala tympani, then through the scala tympani with a sort of a perilymphatic vent with a posterior semicircular canal fenestration. And then lastly, we delivered vector into the endolymphatic sac to see how this would fare, presumably accessing more of like a scala media compartment. And so when we looked at our data, the first thing we realized was that the round window with canal fenestration was the only method that demonstrated transduction through 3.5 turns of the guinea pig cochlea. And this is consistent with what's been found with AAV in mouse models. And so here you can see the reporter protein in green, and then nuclei in blue, and transduction through all the turns of the guinea pig cochlea. And we can look at a comparison of the three different approaches and see that the round window alone method did show transduction in the cochlea. However, it only transduced up to the first 2.5 turns. And with the endolymphatic sac approach, we did not see any transduction in the cochlea. Then when we took a closer look at what was happening on a cellular level, we found that for both round window approaches, so that's with fenestration and without fenestration, we saw transduction within the organ of Corti. And here you can see nuclei in blue, vector in green, and then hair cells in red, and then neurons in white. And the transduction we saw was limited to pillar cells and diater cells within the organ of Corti, which is consistent with what you would expect with the receptor that is used by the helper-dependent adenovirus type 5 particle. But the most important thing about this was that we found even with this larger particle, we were actually able to cross the basilar membrane and enter the organ of Corti to transduce these cells, which was a notable finding for our study. Then when we looked at the medullary region, we also found an interesting finding, which was glial cell transduction was pretty robust, but only if we did the round window with canal fenestration delivery. And this is notable because it would suggest that the entry of the vector into this kind of more central medullar region was through possibly distal aspects, like the helicotrema, and sort of by the apex of the cochlea. And then when we look at the endolymphatic sac delivery approach, as I mentioned, we did not see any transduction with the in the cochlea with this delivery method. However, we did see transduction of marginal hair cells of the vestibule and along the saccule. So again, we can see here it's nuclei in blue, that our vectors in green, and then our hair cells are in red. And so this suggests that this might be a potential avenue to deliver gene therapy with this vector to the vestibular organ. And so in conclusion, with this study, we were able to delineate where our vector travels in the guinea pig cochlea. And we found that with the round window alone method, we were able to access the cells within the organ of Corti. Round window with canal fenestration gave us the added advantage of also accessing cells along the medullus and glial cells. And then lastly, although the endolymphatic sac approach did not show us any transduction within the organ of Corti or the medullus, we did see transduction of vestibular hair cells. And so looking at where we're at and where we're going, the first thing we wanted to do is what we were able to accomplish here, which was establish delivery and vector distribution in a small animal model. And looking forward, we would then be able to take this information to go on to test HDAD of different serotypes and then perhaps different promoter elements before going on to large animal models and human trials. So with this, I would like to thank my co-PIs, Dr. Young and Dr. Hansen, also my co-resident, Osama, who has been a huge asset with COVID and some of those delays and a huge partner in small animal otology, and the members of the Hansen lab listed here. And of course, our funding from ANS was instrumental in making all of this happen. Thank you.
Thank you, Dr. Correa. It seems that we ran out of time for questions. However, participants can place their questions in the chat, and they will be answered by the speakers.